Good afternoon, everyone, and welcome to the Poultry Health Research Network Center. It is my pleasure to introduce Dr. Nagy, even though she doesn't really need any introduction. But I can tell you all the good things about Dr. Nagy. She has received her DVM and PhD degrees in Hungary, and she came to Guelph to do a postdoc, I believe, with Dr. Peter Derouge.
Mm-hmm. Yeah.
And she's been with the Department of former VMI and Department of Pathobiology since 1988, and she rose to the rank of full professor, and she's currently a full professor in OBC. And in addition to that, Dr. Nagy has received a significant number of awards and honors, including the Doctor of Sciences degree from the Hungarian Academy of Sciences in 2001, and in 2004 she was granted the distinction of honorary professor of the Faculty of Veterinary Sciences, Szent István University, Budapest, Hungary. And I'm sure that I mispronounced that, but I hope that you...
St. Stephen.
That's what she said. And she became a member of the Hungarian Academy of Sciences in 2010. Dr. Nagy's research interests are in animal viral diseases with an emphasis on avian viruses and development of the next generation of viral vaccines. She has published a significant number of papers, 130 to be exact, in high-quality journals. She has authored or co-authored several book chapters, and she has presented in excess of 190 presentations in international and national conferences. At the moment, Dr. Nagy is the coordinator of the poultry program at the Ontario Ministry of Agriculture, Food and Rural Affairs and the University of Guelph. She is also the editor for the Canadian Veterinary Research Journal, Canadian Journal of Vet Research. I've got it.
Yes.
And a member of the editorial board for the online veterinary journal of Veterinary Sciences tomorrow. She is serving on the International Advisory Board of the online FAO established veterinary biotechnology and epidemiology network for Central and Eastern Europe. And at the University of Guelph, as many of you know, and probably some of you have already taken her courses, she teaches viral diseases and virology, so viral diseases and molecular biology of viruses for grad students, and also virology for the DVM network. So without further ado, Eva, the floor is all yours.
Okay, thank you very much, Cheyenne. So since there were already 2 seminars from my lab this year, I don't know what news I can tell you, or new items, and hopefully it's going to be even a 3rd one later on this year. So to begin with, but thank you all for coming and for the introduction, Cheyenne. This is just since Ravi asked me about this building. This is the beautiful building of the Hungarian Academy of Sciences that was built about 150, 60 years ago, and looks the same building. So it's really great links for me to this this building. So I will talk, my research is focusing on fall adenoviruses, and we're looking at the molecular biology of these viruses, immunity to these viruses, pathogenesis of these viruses, and the focus today is going to be development of fall adenoviruses as vectors. So I will talk a little bit about the newest taxonomy of these viruses, and I hope that Grant Maxey would have been here to learn of this. And I talk a little bit about diseases and genomes and functional studies as they relate to the generation of vaccine vector. Adenoviruses can infect a wide variety of individuals and animals, so different species and even humans. And I just put up well, I guess some people recognize him, right? So, and actually, so that's one of the newest member or species of the adenoviruses is a skunk adenovirus that we just published not long ago. Here is the phylogenetic tree of adenoviruses based on the hexon amino acid sequences. It shows the variety of these viruses, and actually the AV adenoviruses that I'm going to talk about are in a well designed or well grouped area. And this is from the 9th report of the ICTV. And this is just to let you know, and if you didn't know so far, that's the last copy of the last version of the printed copy of the virus taxonomy. It's going to be available online from now on. So in the family Adenoviridae, there are 5 genera. One of the most, the largest one is the genus Mastadenovirus, genus AV adenovirus, and so on and so on. Some of the adenoviruses that are infecting avian species, the majority are in the genus AV adenoviruses. We have the egg drop syndrome virus in the genus Adenovirus, and the hemorrhagic enteritis virus of Turkey is a genus adenoviruses. Okay, so what's new about this taxonomy is that these viruses are not fall adenoviruses any longer, but fall AV adenoviruses. So all of them, it's going to be, it's like going to be should be a shift in my change to say that fall AV adenoviruses instead of fall adenoviruses. However, the abbreviation will remain the same for these viruses. And like that used to be another virus I renamed very recently. Yeah, that was Turkey, but that was another name, and I added that used to be Turkey adenovirus type 3. So anyway, just follow up the ICTV. In each species, there are different serotypes of fall adenoviruses. They are 1 to 12 because there is 8A and 8B of these viruses. There are three major diseases related to or caused by fall adenoviruses. The most important from them is the inclusion body hepatitis in Canada. Hepatitis hydropericardium syndrome is not seen in Canada, but widespread worldwide. And the upcoming new clinical disease, what we see is the gizzard erosion, ulceration, the GEU. That's reported from other countries. For example, it's very hot area in Europe, and also in Japan and Korea, cases were reported. Other diseases like inclusion body tracheitis, inclusion body ventriculitis, quail bronchitis, also reported as caused by fall adenoviruses. So it's all what you want to know about inclusion body hepatitis. As I mentioned, this is an important disease in Canada, although since the introduction of vaccination, fewer cases are seen in these days, and the vaccine is the autogenous vaccine, which is serotype 8. Inclusion body hepatitis is a disease of broilers and can be seen, and it's seen worldwide. In Canada, mainly serotype 2, 6, 7, 8, and 11, and are causing the disease. And the most virulent is probably the serotype 8 virus. This is a coming back issue all the time, whether IBH or the fall adenoviruses are primary causative agent or is associated with other viruses or other diseases. That's mainly with chicken anemia virus or infectious bursar disease virus that are immunosuppressive viruses. What we see is a newer for the IBH epidemiology that we see the disease is younger birds and the mortality is higher. Clinical signs are non-characteristic. Sometimes you just see depressed bird which dies suddenly. Post-mortem characteristic clinical signs, the liver enlarged, pale, friable, hemorrhages, and of course the characteristic inclusion bodies in the nuclei of infected cells can be seen. Important thing about the fall avian adenoviruses that they can be isolated from healthy and diseased birds. So therefore, it's actually many different serotypes. Antibodies can be identified easily. However, these viruses, and actually the disease, is not linked to one virus species. And the disease is caused by viruses belonging to different serotypes, so there's no species, no serotype association. And within the serotype, there are the pathogenic and non-pathogenic viruses, and that's even complicating the diagnostic issues. And importantly, there is no virulence, there is no marker for virulence in these viruses, so there's no specific particular sequences that one can pinpoint out and that can be used in diagnostic methods to identify the virulent fall adenoviruses. Way back, now it's almost 20 years, there was a paper from Australia that published that it's actually the fiber is responsible for the virulence of these viruses. And since it was published in Journal of Virology, everybody took it and considered that must be true. In Germany showed that one amino acid substitution in the fiber 2 sequences can actually change the virulence of the viruses. However, what he didn't or they didn't do is actually they didn't do the revertant viruses. So that's it's really the value of this results. And moreover, that though we sequenced an FADV4 virus, we didn't find those changes what Michael Haas was talking about that. So of course we were interested in the virulence of these viruses as well. So we took FADV8 and FADV11 viruses, these are those serotypes, taking pathogenic and non-pathogenic viruses for both. And Helena compared the sequences, and she didn't find any association with particular sequences on the fiber. So the question is still out there, what part of the genome is responsible for the virulence of these viruses? Hydropericardium syndrome is what I show here, a characteristic picture, is caused by serotype 4 viruses. Although there is a report that FADV8 can cause hydropericardium syndrome as well. There is vaccination, although somewhat primitive vaccines. It's homogenous that use of the livers that used as for vaccine. I don't want to spend more time on that. Now, can we consider IBH as an emerging disease? Maybe. It was first described in 1957, so it's not a new disease. However, and hydropericardium syndrome in 1987. However, until the 2000 more, that was the kind of a switch when we saw high number of cases all over the world and what reported practically from everywhere. So maybe the reason for that is more sophisticated diagnostic methods, more awareness among producers and veterinarians about this disease. So the gizzard erosion is mainly seen in Europe, and this is Japan, and is seen in Korea. So the adenoviruses are non-enveloped viruses. They have an icosahedral symmetry, and they have characteristic protrusions of the fibers from the icosahedrons, medium to large viruses, and it doesn't matter which genus we are looking at. The morphology, structure is the very same for these viruses, and the proteins on the surfaces, and some of the inside as well conserved, but not for all species or all genera. The genome for all adenoviruses is linear double-stranded DNA. It has inverted terminal repeat at both ends, and there is a protein attached to the double-stranded DNA, which plays a role in viral DNA replication. The most studied virus is the human adenoviruses, and the genome size of this virus is about 35 kb. The smallest one is a Turkey adenovirus type 3. This is 26 kb only, and the fall adenoviruses are a lot larger than the human adenoviruses. So I switch from the disease now to the vector development. Human adenoviruses are used and developed many years ago as vectors and gene therapy vectors. About 10 years after the human adenoviruses, the animal viruses were considered as well, mainly the bovine adenoviruses and the porcine adenoviruses. Bovine adenoviruses was developed at Vido. They worked a lot with that and still working on these viruses. The porcine adenoviruses, actually we worked a lot here, goes back to Dr. Derbyshire, and we were able to generate recombinant porcine adenoviruses, which is actually licensed to a vaccine company now, that actually the construct. So soon after we had this success with the animal adenoviruses, we thought, why not to use the fall adenoviruses as well as the vectors? So here are just the steps, and when the idea came, we didn't know too much about the fall adenoviruses. So just here are the important steps, what we had to do. First of all, select a virus and a strain, what we wanted to use as a vector, and that was the fall adenovirus 9 in the species D. And this virus is non-pathogenic, and it's rather rarely occurs in poultry. So what we had to start with, sequence this virus, generate an infectious clone, find insertion sites for the foreign gene, create that recombinant viruses, and once it's there, look at the immunity against these viruses. And once we have this virus, a recombinant virus, of course, there is always room for improvement. So I go through all of those steps, but before that, I just want to show you a list of the advantages why fall adenoviruses are good as vectors. Actually, you can read yourself. They are stable. They are growing to high titers. There's a variety of serotypes. And actually, that will circumvent the usage of the pre-existing immunity in humans. So what can consider these viruses as an alternative for human gene therapy purposes as well. The first fall adenovirus sequence was the Cilovirus fall adenovirus 1 in 1996, and that followed by FADV9 in 2000 by us, FADV4, FADV8, and that was done by our laboratory. Of course, since then, the deep sequencing possibilities, many more virus strains and isolates are being sequenced. What you see here is the blue arrows indicating genes and open reading frames for these viruses that are conserved among these viruses and among the genera of fall adenoviruses. You see the huge area on the left, on the right, and these are the early genes and early regions that do not show any homology with the mammalian adenoviruses. And what can actually, or you notice here, that the sizes of the fall adenoviruses, as I mentioned before, that's about 10 kb larger. So what are those genes and ORFs are doing for fall adenoviruses? Because the structurals are, the genes and proteins are the same, and it's conserved. And I don't have time to talk about that. Davor also did a transcriptional map of these viruses, or actually the FADV9. And why is that important? Because extensive splicing is, talking about food, right? At just about lunchtime. So it's splicing occurs, that's what you see here. And what unfortunately, again, I don't want to go into the detail of the molecular biology of these viruses. What he found is a tandem repeat region at the right end. And although we call it the E1 region, this region doesn't show any homology to the mammalian adenoviruses. And there was a huge disappointment for us because it's a E3 region, early region 3, between a P8 and the fiber protein and a gene. This region was used for foreign gene insertion for the mammalian and the bovine and the porcine adenoviruses. So, okay, so this is out. So what can we do if we want to use this virus as a vector? And here's just a summary of the unique features of the fall adenovirus genomes. So they are large. There are 6 regions of transcriptional units, and importantly here is no regions homologous to early region 1, 3, and 4, although we call it E1, but they are not equivalent for that. And repeat regions and no other functional genes present. So as I mentioned, that is going to be, so here just a comparison of mastodonviruses and avian adenoviruses, beautiful conservation of the genes, except that it's actually, we don't have this E3 region as I mentioned earlier. And left and the right ends are coding for the genus-specific genes. So one of the idea was that are these, or at least some of these genes, are dispensable or not for virus replication. Of course, the first obvious choice was that maybe we can target this tandem repeat too. But it didn't prove to be stable one. So, and what we had, and it's actually, I think we are still the only lab that has an infectious clone for fall adenoviruses. And that was generated for FADV9 by Davor. And we used this FADMID system to identify regions on the genome that could be deleted and the virus is still replicating. And here came Juan Carlos Corredor, and he explored and studied the left end of the genome and made different deletions and identifying parts that can be deleted. That was really interesting and surprising because for the human adenoviruses, these regions are non-dispensable, so they are essential for virus replication. The little virions are indicating that with even this deletion, the structure gave infectious particles. So we were choosing one of the reasonable deleted virus. It's called Delta 4 for our further work. And just showing that this virus grows to the high titer in compared to the wild type virus, and there is no morphological difference in plaques when we examine them. Of course, the next question was what happens in a chicken when we delete a particular part of the genome from the virus. Interesting was that, of course, there was a rescued virus made as well. So the virus shedding practically disappeared, diminished from chickens that were inoculated from the deleted, the Delta 4 viruses. And we looked at the antibody response, although that was a little bit lower in the deleted Delta 4 virus than in the what's so-called wild type virus and the rescued virus, there was still very nice and good immune response. So that means that we can work this area for foreign gene insertion, and that's what Juan Carlos tried next, took the EGFP and put it into this region without deletion, and this is the inserted gene, or into deleted gene, deleted area. And as you can see here in the chicken hepatoma cells and the liver cells and the QT cells, very nice expression of this EGFP. Moreover, not only the chicken cells, but mammalian cells were transduced as well by these viruses, the rabbit kidney and some of the human virus or cell lines as well. Again, immunized chickens with this now recombinant virus, although we are using a reporter gene, the question was, is this the foreign gene going to be recognized by the chicken or not? Here are the ELISA results and using FADV9 against the virus. And if you see here that the blue, it indicates the antibody titer against the EGFP. So that means that we were able to generate a recombinant virus that is replicating in chicken, inducing immunity. So we had to move on and have the next step. Take a useful gene and the protein that is going to be expressed by this vector. Now we can call it a vector. And we considered and we took the hemagglutinin of the influenza virus and put it into this delta 4 left-hand region. And okay, this is the important picture, Western blot here, and that was DMV who did this work and showing that the HA is expressed in this construct, both in the right and leftward orientation, which is believed that some of them is better expressing than the other one. Also, we studied, because there's always a question with insertion of foreign genes, whether this foreign gene is tolerated by the virus in the long term or not. So went up to 10 passages. I'm not showing all of this. And yes, indeed, this hemagglutinin, the whole hemagglutinin was inserted, gave us, it's actually a stable virus. Now I'm showing, and this is the time when I couldn't go further, or we couldn't go further here in Guelph because we couldn't do the challenge studies. And we'll have industrial vaccine company partner. It's in Mexico. The company name is Avimax, and they did the challenge study with our and FADV9 HA vaccine with different combination. In using NOV, one-day inoculation with different permutation of the possibilities, and they looked at immune response. So what you see is actually the FADV H5 inoculated day one. It's providing 100% protection, and of course they have other viruses as well, so they did testing as well. So that's really good, and hopefully this product and vaccine is going to be on the market soon, later this year or very early next year. And Pei was really busy in the lab, and he was doing, of course, as a basic science, adding other different genes as well to this region. Hemagglutinin neuraminidase from Newcastle disease virus, again, both orientation, and he has a construct HNHA, that's influenza, and using different promoter and iris from a chicken virus. So there's a lot more virus to be tested, and hopefully we're going to do that as well. So that was the left end, but if we want to do, so that's already, we have a polyvalent vaccine, but what about the right end? Can we use the right end as well of this virus? So what Pei did, applying a new recombination technique for his system, systematically deleting some of these ORFs. But importantly, he generated a relatively large ORF here, the TR2, ORF17, and ORF11 are deleted from this, this region. And here, he put the green fluorescent EGFP into this region. It's a very tiny little area here, and these are just the very fine gels of his really good work. And that's what you can see here, that this, the EGFP is expressed, so this area can be used and the virus is viable. But that wasn't enough for us, so we wanted to show actually indeed that both areas can be used, the left and the right end of the genome. So what you see here, and this is the final product here, on the left hand into this delta 4, the M cherry was introduced, and on the right, where we have this delta TR to 1711 deletion, the EGFP is inserted. And Pei is working very hard on that, titrating, and the next step here is going to put useful genes into the region and publish these results, Pei, right? That would be, or tested further. Of course, we can go further optimizing our foreign gene expression, getting these super duper vector and viruses. And you, some of you remember these slides that James worked when using different promoters. In our previous studies, we were using the human cytomegalovirus promoter, and perhaps this is not the best promoter for the chicken system. So we tried in a different construct, what James tried, the CMV, the CAG, EF1 alpha, and the L2R from pox virus, and also using enhancer elements as well. According to the transient expression of EGFP, because that was his system, CMV seems to be the strongest promoter that provided highest level of EGFP in his system. Of course, that was not enough for us because that's still a transient system. So he generated recombinant viruses and with the different promoters and elements, and looked at the titer of these viruses, and it just showed that the EGFP is expressed really nicely. Surprisingly, when he measured the EGFP generated by these viruses, he found that the CAG was the better promoter than the CMV. So it shows again that it's actually the cells or the transient expression system, as much as we like that, not necessarily the best to measure activities. What he did after this and further that we looked at chickens immunized, and we are still working on this data. Because I was told not to talk more than 30 minutes. So here I have the conclusions of my talk, that Adenoviridae is a heterologous family, and they have unique features. Some of the ORFs at the left and on the genome are non-essential, but they are important in virus-host interaction. We have shown, and I hope I convinced you, that FADV9 is a suitable virus as a base vector. Whether the abolished viral shedding is an advantage or disadvantage is out for the market, and I think that the producers can discuss and talk about that. It depends on what virus we are talking about. I think it could be a huge advantage as well. We can put large foreign genes, or the capacity is really large because we are deleting 3.7 kb on the right, 2.4 kb on the left, plus the additional capacity of these viruses. So we are getting up to 10 kb. And these viruses are stable, and we can generate these viruses to high titers. And importantly, for shown that the protein that is expressed by these viruses can induce protective immunity against, in this case, influenza virus. That will open the lead for us that we can make monovalent vaccine, and because of the different sites, we can make polyvalent vaccines as well, taking genes from viruses, and not only from viruses, but perhaps from bacteria. I always toyed with the idea to take Clostridium genes and put into this vector system. So we are very close to an application for poultry. And as I mentioned, adenoviruses will can infect non-avian cells as well, so transduce these cells. So whether we can use and utilize these viruses as gene therapy vectors in the future, something interesting to study. I want to acknowledge the many people who worked in my lab, and they did a fantastic work, and I didn't even talk about Lee's work of many years as functional analysis. Avimax is the company in Mexico who is doing hopefully going to market this vaccine. My funding agencies, and I want to say thank you to everyone, and I just hope that nobody left out from this list. I should take a more recent picture from the lab, because these are the work study students. They are already gone, and Brian is in Winnipeg. Hopefully we see him before Christmas. And but I want to close my talk with this last slide, that we really busily work in the lab doing basic discovery research and publishing sequences and promoter strengths. What we hope, actually, not all of the work what we do, but hopefully some of it is going to give us that we can test in animals that we can use, so at least go to the proof of concept. And if we are lucky, and very often or sometimes we are, not all the time, the products can be applied to the industry, and there will be practical application of that very hard work in the research lab. And I think with this, I stop, and I would be happy to answer your questions. Thank you.
Well, thank you very much, Eva. And I don't really think that I need to conduct the meeting today, so Eva is at your disposal. Any questions?
And I'm glad that the people are here. Some of the people are here in this room who did the job, so they can answer some of the difficult questions if you have some.
So perhaps, Eva, I can ask you a simple question, and perhaps a loaded question. So as you know, Canadian Food Inspection Agency doesn't want us to do vaccination of poultry in Canada, even though somewhere in their guidelines they refer to vaccination. And you and I have collaborated on developing vaccines for avian influenza. What are the chances that we are going to see vaccination not as a standalone methodology, but as a complementary approach towards control of the virus, in addition to biosecurity, in addition to stamping out policies and so forth in Canada? Obviously in Mexico it's been a common practice, and so was in many other parts of the world.
Yeah, so if you look at, there is always a market. There will be a market for a vaccine because China is vaccinating as well. So that's really good. And South America is vaccinating. And I think there are some policy changes or will be policy changes in North America as well. And I think that especially in light of this huge outbreak in the United States this year, and I think there is actually, I don't remember, one of the vaccine is approved and will be, or it is stockpiled for potential use, because I believe and I think that's not acceptable for the public that millions of birds are euthanized, and not even the contact birds, the quarantine birds. And I think that this is the policy. The Netherlands did the same thing, doing ring vaccination. If it's one individual farm, maybe, okay, that's a stamping out, but for millions, that's not acceptable, and the public is not going to accept this. So I think that we are moving towards vaccination. And you know from your own work that because another important thing is when there is an outbreak, that to reduce the shedding, virus shedding from the infected birds. So that's what another important thing, that it's actually how we can, because the amount of virus present in these buildings or when it's an outbreak is tremendous. So if we can vaccinate, reduce actually, whether it's going to be an H9 virus or H6, or I don't know, pick and choose, or it's a genetically engineered vaccine, that's a different thing. But reduce the virus load in the environment, and that's very, very critical. I was at the World Veterinary Poultry Association Congress in Cape Town, and of course every vaccine company wants to get your attention and as a customer, and SIVA has a herpes virus based influenza vaccine, and they are showing beautiful results of that. So it actually was like goes to the public talking about why are we killing millions of, what was the number? It's close to 60 million birds, right, in the States. We're losing that much if we actually do this ring vaccination in areas. So, and showing the reduction of the virus, and not necessarily we want to move away from the vaccination, but in emergency situation, I would recommend, yes, do and vaccinate.
Well, this is obviously music to my ears. And the fact is, it's quite known that the virus that hit the US, the H5N2 highly pathogenic that hit the US and also hit parts of Ontario and BC, it wasn't really a virus that we would have expected to see in terms of where it came from, and also in terms of some of its characteristics, because I read in some of the documents that are now becoming available, some of those poultry farms had actually undergone very extensive biosecurity audits a few weeks prior to the outbreak. So it meant that they were quite biosecure, but for some reason the virus managed to identify if the virus was smart enough, and apparently it was smart enough.
So the virus went viral. Yeah, that was it. So.
And it identified, all the caveats in their biosecurity schemes, and it managed to penetrate through those biosecurity measures.
Yeah.
Yes, Bob.
Thank you very much. My question is about hydropericardium syndrome caused by this disease, by this virus.
Mm-hmm.
Actually, the age range of this disease that you mentioned is exactly the same age range of normally occur ascites and hydropericardium syndrome, which occurs because of the heart failure. So how we can discriminate between these two? And also we know that the virus, as we said, can be isolated from disease and also healthy birds.
Okay, so the question was, how do we differentiate hydropericardium syndrome, the virus-caused hydropericardium syndrome, from the hydropericardium syndrome, which is normally could occur in meat-type birds. This is where it comes back, and it doesn't matter how many times or who you talk to, it is going to be, the question goes back, is it the primary agent, or it's going to be, or there is other causes in the disease. And I think with many cases, and now I'm doing more reading about the gizzard erosions, what we see is actually there might be something else all the time. So to answer your question, if the virus is there, that might play a role, okay? But there is it goes back. There is no genetic marker to say, even it's a virus there, whether that cause or can cause. You can go take SPF chickens and inoculate, and maybe that would be, because that would really kill the virulent FADV4, would kill 10 days old SPF chickens.
Because it is interesting that this virus type is only causing the disease in broilers at 3 to 6 weeks of age, which is normal for hydropericardium and ascites syndrome, and not in, for example, layer pullet chickens.
That's true. I didn't emphasize this one because what we see, there are a few cases in layers as well, outbreaks, but not as serious, or the virus can be isolated. So is it something to do with the genetics of those birds? Or is it something to do with the physiology of those birds? Or is it something, what happens in the liver of these chickens? Well, there are two, actually, I had the slides on that by 6 weeks of age or 7 weeks of age, we have a 2 kilogram chicken. Am I correct, Keith? Yeah, more or less. Yeah. Okay. So, but that's a tremendous growth. And when I don't want to go back, when it was, since 57, the weight of a broiler chicken is 4 times higher. It's quadrupled. We didn't have, the virus was there, but we didn't have IBH in 57 or the 60s. So we started to actually see the IBH in broiler chickens when this huge increase in weight gain was realized. But, okay, and there would be interesting to look at. We started to look at it, but it's difficult to take the indigenous birds, which we don't have that many, and look at their response to the disease. How did they go? So there's really something to do with the virus or with the birds.
So maybe the physiological defects in broiler chickens predispose the condition for this virus to act.
You said that. Okay, yeah, which is actually, I incline to agree with this one. So that's another, I guess in a situation like that, the change in the ammonia concentration or in feed change or coming a coccidia, that could actually cause a disease.
My question is regarding the shading, for example. You mentioned when you delete the left end, it's a kind of single cycle where you don't see any shading. And also you plan to have a polyvalent vaccine just to target the other end. In such scenario, what would be the nature of the virus? If you insert, for example, HA on the left end and, for example, HN on the right end, does this virus still replicate the shades?
Okay, yes. That's, I think it's going to replicate the, oh, sorry. To repeat the question that was, so that what will happen to virus shedding, we have the double insertion virus. And both, but different, yeah, we're having different, you don't want to put the same because that would be a competition anyway, so now, but what will be with the virus shedding, and well, first of all, I think, is this virus going to replicate in the chicken, right? Or, yeah, the virus will replicate. I think in the chicken, it replicates well in the cell culture, so it's going to replicate in the chicken. Now, I didn't have a time to talk about that at all, but whenever we have a chicken experiment, we always determine at least 3 or 4 chicken experiments that I didn't talk about that, if it's not more, that we always determine the amount of DNA, so look at virus replication by quantitative real-time PCR in different tissues. So you look at the liver, the secret tonsil, the bursa fabricius, as well to see where the virus is. And yes, there is virus replication. It's somewhat lower, but not much lower, the virus replication. So why is this? The virus is not shedding? That's we will see. That's an interesting question. So I don't think that's going to be the 2 genes are going to impair the virus, or it's actually the recombinant virus.
In the vaccine trial in the AVMax, the Mexican company, was it single immunization or multiple immunization? And what are the route of immunization there?
They were, that's numerous vaccine trials, and that was presented at the influenza meeting in the spring. So that they tried to get single, or it's actually, immediately when you have to give another one, that's not going to be too economical. One way it could go, they'll have an NOV, and you would get later on one day. But they have a magical adjuvant, apparently, what they are using. I don't know, they're not talking about that, but it's definitely, the goal is to have single immunization for the broilers, or it's going to be, if it's going to be for the breeders, no, that's going to be a different scenario, and how to give this vaccine, that's another one, because another question. They gave the vaccine in OVO, and in day 1, that was subcutaneous. But we showed that if you give orally the virus, that's going to induce immunity as well. And that was the advice of Dr. Sharif to try this experiment. Mm-hmm.
Yeah. I was curious to know if given spray, like the virus can multiply in the respiratory.
We haven't done that because I don't, well, it could be, that could be done, but it's usually not given through the conjunctiva.
If you have time, we have questions from Dr. Wilkie.
Oh yeah, I will stay here if somebody wants to.
Maybe I can go ahead with Dr. Wilkie and then keep the floor to Shireen for questions. So,
Oh, okay.
Duration.
Okay, so what about the duration of the immune response? We usually keep, it depends on the number of vaccination or inoculation. And Juan Carlos had an experiment, and we kept for how many, 7 weeks, the chickens? 7 weeks, and there was still a good immune response right back. So it's good. If it's repeated, it's going to be sustained. But normally it comes down. And it's going to be, that's what from the field, so that's going to,
So it's not a problem.
Yes, probably not. The problem with the immunity and the titer, if it's any, going to be that this different serotype and cross protection, cross reaction, when it's going to be. So that's something need to be studied and looked at.
Shireen?
She has just answered my question. It was in regard to cross-protection studies for the FABP9 containing the H5 insert. So I guess new cross-protection studies have been done.
What, regarding the vector or the influenza or the H? No, there wasn't 100% protection. But one of the things, it's almost like when I meant the cross protection or cross neutralization was for the vector, because if there is antibody for the vector, it's not necessarily is going to take.
Actually meant in terms of the H5 itself.
Mm-hmm. Yeah.
Well, wonderful presentation. Thank you very much, Eva. I'm just
Okay.
going to conclude the meeting today, but the whole purpose of today is really to mingle and say hello to one another and also enjoy some coffee and donuts. And I think we've got lots of tidbits left, so by all means, thank you very much, everyone. And thank you very much.
Thank you.